Because you and your classmates are going to be getting to the point where you need to take notes at different times, I thought that I would make a video that shows the steps of transcription and translation with the PowerPoint notes. I would like you to add these to your notes. Some of you might already have, like 6th hour already has this first slide completed, so you can just go through this real quickly, but then you're going to need to join up with the steps as soon as transcription is done. Okay, so remember that protein synthesis has 2 steps to it. It has transcription, and then the second step is going to be translation. Transcription is what's going to take you from DNA to RNA. Okay, so that process is transcription. The next step, the second step in making a protein is called translation, and that's going to take you from RNA to protein. Okay, so there's 3 different steps, and this is called the central dogma that's going to be involved in the production of proteins, going from DNA to RNA to protein. But there's 2 processes that are going to be involved with it. The first one is transcription. We focused on that yesterday with the models, and then the second step is kind of today's focus, although I know we have to finish up transcription first. But the second step is called translation. Okay, so now let's go ahead and get through those notes. Okay, so those people that do not have these notes, you got to go through this will be review then, because we did it with the models yesterday. In order to do transcription, the purpose of this, as was just mentioned, is to synthesize RNA, and it's going to be using the instructions that are encoded in the DNA. So the DNA serves as a template to produce the RNA. In order to do this, we have steps. A portion of the DNA molecule is going to unzip. We don't unzip the whole thing. We only need to make 1 protein, and so that's 1 page out of our 20,000 page recipe book that DNA is. So we don't need to unzip the whole thing and rewrite the whole 20,000 page book if we just want to make the recipe that's only on 1 page. So only a little bit of the DNA molecule is going to unzip, which means that the hydrogen bonds between the bases are broken. There's an enzyme that's going to do that. Then, once you have exposed bases, messenger RNA nucleotides are going to use one of the 2 sides of the DNA molecule, called the coding side or the template side. That side is going to be read, and you're going to produce a strand of messenger RNA that is complementary to that. I have this strand of messenger RNA written in yellow because that is our product of transcription. Messenger RNA is produced because it can leave the nucleus. Remember, DNA is unable to leave the nucleus, and so we have to have this go-between, which is messenger RNA. It's kind of like the note card that contains that information, or those single pages in the video that he was throwing out into the cytoplasm C. So that leaves the nucleus and heads on out to the cytoplasm where the ribosomes are for translation. The DNA molecule is not destroyed by this process. It can go back together again and be reused over and over and over and over again. So the hydrogen bonds between the bases of the DNA would be reformed, so I'm saying it's rezipped. Okay, this just goes through those steps. So this is our starting gene, the DNA of a gene. You have the promoter region, that's at the beginning of the gene, and you got your enzyme there that is going to be recognizing the promoter. Then here you can see how a little bit of the DNA molecule has separated from its complementary side. And we're starting to build an RNA transcript complementary to the template strand. So in this case, our template strand happens to be the bottom strand. It could either be the bottom or the top. And this is showing what's called elongation, where we're continuing to add new nucleotides to the messenger RNA that are complementary to a section of DNA. And when we're finished, we have our completed messenger RNA. And notice the DNA has gone back together again so that it could just be reused over and over again. So that's the process of transcription. This is a different illustration kind of showing the same thing where you have the DNA separated out and then you're building the messenger RNA that's complementary to it. It shows the base pairs there, A's to C's, and then, A's to U's, and then C's to G's, of course. Okay, so in order to understand translation, we also have to talk about a second type of RNA. There are several types of RNA, but we're only dealing with two of them. The second type of RNA is called tRNA. And when I think of tRNA, I think of trucks. Okay, so it's like truck RNA. And what trucks do is they carry a load of something from one place to another place. And that's what these are doing, is they are transferring the important ingredients that are necessary to build our protein to the kitchens, okay, to the ribosomes where they're going to be put together. In order to put the ingredients together in the right order, there's two really important specific regions of the RNA molecule. All right, so it's going to be putting them together in the correct position. That amino acid sequence is the only difference between insulin and fibrin and hemoglobin and any of the other proteins that you guys did for your protein poster. The only difference has to do with which amino acids are put where. Amino acids, they are the building blocks to make proteins. So that's what proteins are made from. That is not what I want. Okay, so there's two specific regions. One of those specific regions is called the anticodon. There's your anticodon. The anticodon is a 3-base combination that is going to match up to the codon, which is the 3-base combination on the messenger RNA. So your anticodon will be complementary to the codon. That's like the transfer RNA having a specific docking position where it needs to bring its amino acid. So it has to line up. It only can insert its amino acid in one position, in one place. It only can deliver it to the codons that its anticodon matches up to. The only other thing that is specific is which amino acid it carries. Now there are 20 different types of amino acids, and those amino acids have to get linked together in a specific order in order to produce, let's say we're making hemoglobin. They have to go together in the instructions, in the order, in the way that the messenger RNA tells us. So there's 20 of these different ingredients. Each transfer RNA only can carry one kind of amino acid. So this particular, let's say that this transfer RNA carries glycine, which is one of the 20 amino acids. It's going to drop glycine off and then go and pick up another glycine, and drop glycine off and go and pick up another glycine. So it's not like it's going to drop off glycine and then go pick up histidine. They're very specific, just like a truck. If it's a milk truck, it's going to go drop off milk and it's going to go pick up more milk and drop off milk. It's never going to go and pick up gasoline sometime or something like that. They're always specific. So only this amino acid is going to be delivered by the transfer RNA that has this specific anticodon. You need those two things to be very particular in order to get your amino acid sequence in a particular order. And so that's how this works. That's how the DNA can code for a particular amino acid sequence. It's because these two things are non-changing. Your anticodon and that particular transfer RNA only carries that particular amino acid. And then this is called the genetic code. You have this on your purple sheet. The genetic code is going to tell you which messenger RNA codons, okay, so these are going to be written in codons. So it tells you which messenger RNA codons are going to code for a single amino acid of the 20 amino acids. So for example, if you had a codon that was UUU, that would tell the ribosome that we need to insert phenylalanine at that position, and that is one of the 20 amino acids. These are just the 3-letter abbreviations, but on your purple sheet they do have the the long names for the amino acids. Okay, so you can take a look at the example here and see if you can figure out what amino acid would be coded for by AAU. Because this genetic code is universal, that means that it's not just for humans. It's a genetic code for all living things. That makes genetic engineering possible, which is some of the most exciting genetic work that's being done today. For example, we can take bacteria and we can insert the gene for human insulin. Now, there's people that need to take this as medicine because they have diabetes and they don't make their own insulin. If they didn't take insulin, they could die. So what we can do is we can put the gene for human insulin into a bacterium, and then the bacteria will make human insulin, which is pretty slick. Not made by a human, made by bacteria, but it is human insulin. The other important thing about the genetic code being universal is because we all work the same way. All living things are operating with this pretty complex code system that it's very unlikely that that could have evolved more than once. So that suggests that there would be a single common ancestor. We all function the same way biochemically. All right, now let's go through the steps of translation. All right, so once you have a messenger RNA code, the second step in producing a protein is translation. So the product here, what we're doing is we're going from RNA to protein. So a protein is our product. The steps. The messenger RNA is going to use the ribosome. So that's the location of translation. That's where we're taking place. And it always is going to slide that messenger RNA in between the two halves of the ribosome. Remember, there's a large subunit and a small subunit. It slides until it finds the start codon, which is AUG. Every single time it starts with that codon. Then transfer RNA is going to deliver the amino acids, but they have the anticodon on them that tells them where their docs are. So it tells them when they can join up and add their amino acid. So that anticodon and codon pairing would end up happening. Amino acids, remember those are the building blocks to make proteins. Those amino acids are going to get linked together by peptide bonds, the type of covalent bond, so it's a strong bond. And it keeps going and keeps going and keeps going until you reach one of the three stop codons. You don't have to memorize the stop codons, but take a look on your genetic code sheet. There are 3 of them. That signals protein's done. We don't need to add any more amino acids. So there's a start codon at the beginning of the sequence that codes for the protein or tells you the amino acid sequence, and a stop codon at the end that signals the end. So this newly formed protein then is going to detach from the ribosome and go do whatever job it's going to do, whether it's keratin or collagen or a motor protein or whatever it does. That protein is completed at that point, or maybe it needs to be processed in the ER in the Golgi apparatus first. But that's going to be kind of the beginning of the structure of your protein. Okay, so here's an illustration that goes through those steps. So once again, remember that here we have a messenger RNA molecule and we have a ribosome. We've got our start codon in the first position there, AUG, and then these are transfer RNAs. Notice the AUG is going to match up with the first one being UAC. That's the anticodon on the transfer RNA, and it's delivering a particular amino acid. That particular one that it always delivers first is methionine. Then, after that, our next step is we would slide over 3 and we would have our next codon here. Whatever that one is, the anticodon would match up to that. So that particular transfer RNA is the only one that could dock up at that spot. And then you would have your second amino acid delivered. You have to form a peptide bond between them to link them together. Once that's done, this transfer RNA is an empty truck, and so it can go pick up another amino acid. And then your next codon is in position. Whichever transfer RNA has the anticodon for that will dock up at that position. And then it'll attach its amino acid until you get this long chain of amino acids. Most proteins are in the neighborhood of several hundred amino acids long, but they can vary really widely. Some of them are a few hundred, some of them can be more than 1,000 amino acids in length.